First question is for Dr. Prentice. You don't use fetal tissue at the Midwest Stem Cell Therapy Center, correct?
No, Congressman.
Has this prohibited you from doing cutting-edge research?
None of the research at the Midwest Stem Cell Therapy Center uses aborted fetal tissue, but they have made great strides in spite of that.
Yep. Can you tell me examples?
Probably because of that.
That's interesting that you say because of it. And there are other centers like yours around the country. Can you explain why, first of all, some of the progress that you've been making, and secondly, why you feel you're being able to make progress at a greater rate than some of the centers who feel they have to use fetal tissue?
Well, the origin of that not using the aborted fetal tissue, frankly, was from the legislature that initially set up the Midwest Stem Cell Therapy Center. And part of the reason was to focus on the more successful adult stem cells and the potential treatments to get this into the clinic faster for the patients. So I mentioned one of the clinical trials. It's a homegrown clinical trial, first of its kind, to treat graft versus host disease using an adult stem cell source. And that clinical trial is still ongoing, so this is very early, but the initial results seem very successful. In association with the Bone Marrow Transplant Center, they have been treating a number of different cancers as well as anemias, including sickle cell anemia. They have also engaged in this stroke study to treat patients who have experienced stroke even years in the past.
Overall, countrywide, has there been more progress with adult stem cells or fetal tissue research?
There's much more.
Not even close, right?
I'm sorry?
Not even close.
Not even close.
Why do you think when it's not even close, there's some people out there who insist? Is there something psychological going on, this apparent love affair with fetal tissue? Why psychologically would people prefer to use that than the more effective adult stem cells?
Well, I think in some cases, as a scientist, we're trained in a certain way, and we may be reluctant to switch to something new, something that, even if we know that it's better and potentially more productive. But, Congressman, I'd like to make a point about that when we do step out, great things can happen. And if I could give you just one brief example, if we look in the stem cell field at the production of these induced pluripotent stem cells, the Japanese scientist Dr. Yamanaka came up with a few years ago. One of his reasons, frankly, was he'd looked through the microscope at an embryo in a colleague's lab and said, you know, we can't see much difference between this and my daughters. We can't keep destroying embryos. There must be a better way to get the same kind of cell. And he came up with these iPS cells, which mimic pretty much everything that an embryonic stem cell can do, and won the 2012 Nobel Prize for it.
Okay. I sometimes wonder when it comes to politicians who are always pushing the fetal tissue, if it becomes because they've been so invested in abortions and they want abortions to be good that they just want to believe that fetal tissue research is superior, and they have this psychological problem. But I don't want to let you alone, Dr. Sanderly. Can you tell us a little bit about reprogramming adult stem cells into pluripotent cells?
Yes. So you can generate what's called induced pluripotent stem cells from somatic cells. So, for example, you could take a skin biopsy from an adult human, and you could isolate the skin cells, and then you could reprogram them. You could give them specific factors that would allow them to differentiate and basically revert back to an embryonic-like state. And then those cells, those pluripotent cells, can then be further reprogrammed into what are called organoids, which are basically simply organs in a dish that can be studied and have comparable, similar cell components to what we see in the fetus.
Can you give me examples of some sort of treatments that are evolving out of this?
So, for example, So probably some of the best examples are when we look at brain development. So there are multiple publications in the literature which show that you can directly take skin fibroblasts from adults. You can actually even purchase these skin fibroblasts from biorepositories such as Coriell. You can make these iPS-derived forebrain organoids, which you can use to study Miller-Decker syndrome. There's actually key publications that have shown that you can take these cells and you can actually make cerebral organoids, and you can actually look specifically at astrocytes, and they have shown these astrocytes actually have functional pathways similar to the adult human cortex. We have other examples, such as Luau and al. From 2016, in which they've generated these organoids to recapitulate many of the features of the mid-fetal human brain. There's other examples of, so for example, Monser et al. From 2017, in which they've taken these brain organoids and they've actually been able to graft them into a mouse and actually look at functional neuronal networks and vascularization, such as blood vessels. So these are all critical studies. And another example, Levine was able to actually take some of these, actually Zhang and al. Were able to actually take these and actually research human developmental brain by recapitulating the medial ganglion organoids and cortical organoids. So there are multiple examples. I could keep talking just about how, and this is just brain development. This is even talking about retinal development, kidney development, and these are so key studies that can be done looking at multiple areas of development in the fetus.
Thank you, and I'd like to
Mr.
thank the chairman for letting me talk.